Yeah, it's for pegylation actually. So they get the bad drugs and they try to pegylate it in a creative manner. And by doing that, they were able to get good PK and good safety profiles. So now the Nectar 214, which is like the main candidate, it's being worked on in like 12 different indications in 12 different kind of cancers. And we are collaborating with BMS, Bristol Myers Squibb, and Eli Lilly. So one molecule is already given to Eli Lilly. This was again
Yeah.
for cancer, and this one we are collaborating with BMS. So it's a huge collaboration. So right now there are like 12 clinical trials going on, and we hope to get
Some
it
are
in the
in
market.
advanced stage. Yeah.
Yeah, some of the advanced stage. Yes, melanoma, RCC, renal cancer, renal cell cancer, and bladder cancer. So all kind of thing, head and neck cancer. So they are exploring almost 20 indication at this point.
Some new agents for autoimmune disorders.
They made something for pain as well, but we didn't. We were not lucky with it. FDA didn't like it. Right now, the political atmosphere is very charged here in the United States, and people don't like opioids. So that's why they kind of put it down.
Right. On Facebook also, I understand. Satya, you are there online? He is arranging for students. We have given it to be like live on our Facebook.
That's really good. You always have been tech savvy. I have to say that. You always utilize it in the best way, and I think in this situation, in the current COVID thing, I think it's very important.
Yeah, we could switch over very quickly to online system.
Yes, sir. We are going live.
Due to some of these, initiatives, some of them were driven by me. My colleagues also were very prompt in adopting this system.
I see.
Dr. Veer Singh has also joined. He is Dean PG.
I see. Hello.
Yeah, Dr. Veer Singh was earlier with Gujarat.
I see.
So he has recently joined as Director Residence in Instruction and Dean PG Studies.
I see. Nice to meet you, sir.
Young faculty
Thank
here.
you. Nice to.
Others have come. Maybe within next 2-3 minutes, more people will join.
Okay.
And
I
then
I can start sharing my screen in a couple of minutes. I was just trying to get in to make sure everything is working. So I jumped on a bit early.
You can briefly introduce your company and your own profile to the audience.
Yeah, sure. Let me start it.
Sir, I will introduce you.
Okay.
So
okay.
it says host disabled participating screen sharing. So I think somebody has to give me access.
I have allowed it. I have allowed it.
Let me see.
You can check here. You can check.
Multiple participants can share simultaneously. Yeah, it's there.
Yeah, it is there. It is there.
Perfect. Thank you. So I guess.
And I also worked at Patel Chest, Delhi.
Yeah, I will go through it. Should I go? Should I start?
Yeah, Ravi,
Yes, sir. Just a second, sir. Dr. Vinay. Dr. Saroj, you are there?
Yes, sir, I am here, sir.
Please just introduce the speaker, then we can start, and then people will join in between. I hand over to Dr. Saroj.
Good evening, sir. Good evening, sir. I welcome our Honorable Vice Chancellor, Dr. Rameshwar Singh, sir, our Director Research, Dr. Vinay Kumar, our DRI cum Dean PG, Dr. Veer Singh, sir, and our eminent expert and speaker, Dr. Vinay Kumar, sir, and all the participants. Very good evening to all of you. Sir, we are having one webinar on the topic immunogenicity assessment of biotherapeutic products. So before proceeding to webinar, I would like to introduce in very brief about the Dr. Vinay Kumar. Sir, he is a safety assessment lead at Nectar Therapeutics, and Francisco, where he is responsible for the safety assessment of various biologics currently in the development for the Immuno Oncology program. He earned a PhD degree in biochemistry from Charles University, which is one of the best university of the Europe. His doctoral thesis are focused on post-translational modifications in soluble recombinant therapeutic proteins secreted by lower eukaryotes, structure and function. He also developed a rat colon cancer model. After finishing his doctoral degree, he worked at Johns Hopkins University, Baltimore, Maryland, and Tulane University, New Orleans, on development of transmission blocking vaccines against malaria and molecular pathology of HIV enteropathy as a postdoctoral fellow. He also studied the impact of cannabinoid epigenomic and mRNA mechanism on HIV disease progression during the rhesus mark model. His research interests are cancer immunotherapy, vaccine development, and molecular pathology of HIV. So now I invite our Honorable Vice Chancellor, sir, for opening remarks. Sir, please.
Speaker for today's webinar, Vinay है? Cut हो गया. अच्छा, Vinay is there. Yeah.
I can hear you.
Yeah. Yeah, you are there. You are out of my screen for some time. So I just wanted to make sure that you are connected. All my senior colleagues, Director Research, Dean PGS, and Dean Student Welfare, Dr. Trivedi, all colleagues and all students who are following us on the Facebook. It's my privilege to introduce and welcome the speaker for today's webinar, Dr. Vinay Kumar. He has a lot of experience in biotherapeutics and especially in the area of new drug development and vaccine development, safety testing of new drugs through immunogenicity. What kind of sometimes we have the allergic reactions to different products. Nowadays we are using protein molecules like molecular and monoclonal antibodies, certain enzymes, and various other agents. So how the industry regulates all the safety parameters and how these are, assessed in the laboratory through different protocols and different methods. This is a very high-tech field in which latest tools of biotechnology, bioinformatics, and clinical trials, they are combined together and they meet the rigorous standards for the release of new drugs. Development of new drugs is a very costly affair. And Nectar Pharma, where Vinay works, is a leading company. I was looking at the turnover, some $5.8 billion in 2019. Vinay can tell more about the present company. And we have been in touch since long when he came to my lab at Karnal as one of the research associates in a project. He was very bright and very cooperative to all the students and staff in the laboratory, and we did some good molecular biology work which related to probiotics and lactic acid bacteria and their properties. So for today's lecture,
I think there is some net issue with the VCSS connection.
devoting more time standing between you and Vinay, I will like to invite Vinay to start his presentation. The attendance will grow, I believe, in due course of time. So Vinay, it's over to you.
Thank you, sir. Thank you very much. I would like to thank Honorable Dr. Rameshwar Singh, whom I have known for a very long time. And I was saying before starting the presentation that, he was my first boss who hired me after my master's back when he was in Karnal. So I had a great time with him, very cooperative, always there to help in any way possible, always encouraging to do bigger things, better things. Thank you very much, sir. It has been a privilege to know you. Respected faculties, dear students, I am Vinay Kumar, and I'm here in San Francisco, California. It's 6:30 a.m. in the morning. Generally, I don't wake up that early, but Dr. Santosh said that that's going to be the most convenient time for the students and faculty. And I said, okay, I'll make an effort. I'll get up. So here I am. So good evening, everybody. It's good morning here. So at present, I'm working as a safety assessment lead at Nectar Therapeutics, and we have 3 main sites. One is here, the main head office is in San Francisco, California. We have a manufacturing facility in Huntsville, Alabama. And then we have a Nectar India office as well in Hyderabad, where they do a lot of PK, pharmacokinetics, and pharmacodynamics sample testing. So we have presence in India as well. So today I'm going to talk a little bit about my job, which is immunogenicity assessment of biotherapeutic products. So we are, Nectar is a pegylated company. So we get some of our one area of expertise is working with interleukins. So as you know, interleukins are highly reactive. They can do immune activation. So that's why they are not widely used. For cancer, IL-2, IL-7, IL-15 has been proved very good as far as the immunotherapy is concerned. So that's where the Nectar's expertise come into the play. So what we do is we take these interleukins, we pegylate them. We put the polyethylene glycol, and this is a very common chemical. It's in shampoo, it's in basically a lot of drugs, it's in ice cream. So it's very safe, very inert. And we couple these proteins, interleukins, highly reactive proteins, in such a manner that it changes pharmacokinetic. So what it does, it changes the size of it, it masks some of the areas. So these proteins who are highly reactive, they become safe, and they could be used in patient treatment without creating troubles. So one of the most important part is safety. When you develop a new molecule, all the regulatory agencies, which is FDA in US, EMA in Europe, European Medicine Agency, the first thing they look is for the safety. Whatever you're trying to bring to the market, or whatever you're trying to test in humans, the first thing is safety. So how are you going to do that? So in today's presentation, I'll go through some of the in silico techniques and some of the lab work, how it is done. And let me start the agenda. So the first part of the presentation is going to be introduction. I will talk about a little bit about my research background before I joined the industry and how I joined the industry and what I do. The second part will be the in silico prediction, which is very important. As Dr. Rameshwar Singh was saying, it's a very costly business, multi-million-dollar investment. So you want to know a lot of things. You want to know as much as possible in very early stages, so you can select the best candidate to go forward. Then I will talk about Excuse me, somebody has to put it on mute, I think. I can hear background noise. Thank you. The regulatory expectation, what does the regulatory agencies want? What are the guidances of FDA and EMA? And then finally, how we develop a method and how we validate it. And I'll go a little bit in details, why we validate it. And then resources and opportunities in regulatory science. It's a very exciting field, and I didn't know about it because I was always in the laboratory. And then when I start going out and exploring, I find it's a fascinating field, and I would like to share some resources. It may be helpful for the students who are looking for jobs and who don't like to be a bench scientist in academia. So that's a good, there are some good opportunities. Please feel free to ask questions whenever you want. Don't mind disturbing me, just say it. So before, I'll go a little bit back. So I got a master's in microbiology in India. And after that, I went to Dr. Rameshwar Singh's lab as a research associate. And we started working on molecular biology of lactobacillus. That was the focus of Dr. Singh's lab in those days in Karnal. After that, I came to Delhi and I work at Patel Just Institute. That's a part of Delhi University. And there I was working on mycobacteria, Mycobacterium tuberculosis, and especially the multi-drug resistant mycobacterium. And we were trying to make some diagnostic tools easy to use in remote areas. Excuse me, there is some background noise. Thank you. So we were trying to make some crude methods in which doesn't require very highly skilled staff or very costly machinery, just to a bunch of basic molecular biology techniques, how you can use them to identify which of the mycobacterium strains are multiple drug resistant versus the sensitive ones. And after that, I went to Charles University, Prague for my doctorate. And there I was working on basically Immuno Oncology. And I worked on natural killer cell biology. We were working on CD69. There is a outer cell signaling part of it. So one of my project focus was to express it in different expression systems. So I was using bacteria, which is a prokaryotic system. Then I was using Pichia pastoris, which is a yeast, and then CHO cells, which are basically eukaryotic expression system. And we were trying to study the effects of glycosylation, because bacteria cannot glycosylate these proteins. It doesn't have that machinery. And CHO cells can do that. And same does the Pichia pastoris. So we were doing a study how the glycosylation help in cell signaling when it comes to CD69. And a part of my project was to develop a colon cancer model, rat colon cancer model. And colon cancer is a big trouble in Europe. And it boils down to basically their diet. They consume a lot of red meat and they consume a lot of beer, a lot of alcohol consumption, very less consumption of fruits and vegetables. So colon cancer cases are very high there. So a lot of efforts was put into the research just to find out the reasons, the causes, and possible compounds to treat it. After finishing my doctorate, I went to Johns Hopkins University in Baltimore. And there I started working on transmission blocking vaccines against malaria. And the reason was I was always interested in public health. It was mycobacteria before, and then it became malaria. And even now, the biggest killer of human being is malaria. So many people are being killed in developing countries, especially in Africa. So that was a really cool concept for me. So that's where I'm going to pick up. I'll give you just a little bit background about the transmission blocking vaccines. So there are three main targets when it comes to malaria. And malaria is caused by Plasmodium falciparum or Plasmodium vivax, depending in which region you are. But the main causal agent is Plasmodium falciparum. And the reproduction happens inside the mosquito gut. So when you think about vaccine targets against malaria, there are three. One is the liver stage in the human liver. And that's when there are symptoms, when you have the fever and chills. So this stage is very popular with the industry because you can always sell it to army. US Army was a big kind of client for this because they send soldiers in conflict areas and they want to have some protection against it. There is no malaria in US, so they don't have any antibodies, they have no protection. In the bloodstream, the blood stage vaccines, and then inside the mosquito gut, that's the transmission blocking vaccine. It was a very public health concept. So I'll just go a little bit details about it. So it all happens when a healthy mosquito, can you see my pointer? May I ask? Anyways, I'll go ahead.
Yes, yes, it is visible.
Okay, so when a healthy mosquito, female Anaphylaxis, take a blood meal on an infected individual. So when the person has malaria and the female Anaphylaxis mosquito take a blood meal, along with the blood, it also take the male and female gametes of Plasmodium falciparum or Plasmodium vivax, depending on in which area you are and which causal agent is prevalent there. So once it take the blood meal inside the mosquito gut, that's where everything happens. So these male and female gametes, they fuse together, they become zygote. Then they go through different developmental stages. They become ookinete, they become oocyst. Once they become oocyst, they kind of blast and they go to salivary gland. So this healthy mosquito who happens to take a blood meal from an infected individual now is infected with malaria. And he's like a flying syringe. He will go around, bite people, and basically spread malaria. So our research focus was, there were some surface antigens on these male and female gametes. There is a family of 6 cysteine proteins. So what we were trying to do, we were trying to express them in E. coli and Pichia pastoris. And then in inoculating mice and monkeys, rhesus macaques, get their serum, get the antibodies, and feed them basically to starved mosquitoes along with the blood. What that will do is these antibodies will stop, they're kind of neutralizing. They will stop the fusion of male and female gametes inside mosquito gut. So it was very difficult to do, that technically very challenging, because you're gonna starve some, first you have to make mosquitoes in the lab, which is a very difficult process and it's hard to understand because outside in the wild, they're always there. But when you want to raise mosquito colony inside the lab, it's very tedious. You have to be very careful with everything. Second thing was the experimental design. So you express your these vaccine targets, then you inoculate your animals, first mice and then monkeys. You get the serum from them, and then you mix it with the blood and feed the hungry mosquitoes. And then you wait for 15 days. And then you dissect every mosquito individually so you can count this oocyst. And you cannot see it by your eyes, so you have to do it under the microscope. So you can imagine if you have to dissect 500 mosquitoes, it was very tedious, extremely tedious job. So we have some very good technical staff who were helping with this. Very tedious, very time taking, very laborious, but extremely important piece of work, given that how many people are killed by malaria. So this particular project was funded by Bill Gates Foundation for the vaccine initiative, MVI PATH, Malaria Vaccine Initiative. So we were able to show very good blocking of transmission, and basically you do it, how many oocysts was formed. If you have functional antibodies against these surface antigens, there will be no oocyst production. So you actually count everything after taking out the salivary glands. So there was very good data in mouse and in rhesus macaques, in monkeys. But when we transfer the technology to private partners under GMP, good manufacturing practice, to make it like a real vaccine, things didn't work out for us. And one reason was that these particular antigens they're cysteine rich. There are a lot of cysteines. So there is a possibility to make several disulfide bonds. And since B cell epitopes are conformational, certain cysteines has to pair in a certain way to get that proper conformation to make the functional antibodies. So in lab, we were making like 10 milligram per liter of these antigens. But when we transfer the technology to the private partners, to the industry, they were able to make it like 60 milligram per liter. So they scaled it up. But something wrong happened in that process, and these vaccine candidates were not functional. they were able to raise antibodies, but those antibodies were not functional. They were not working to block this fertilization. So that project has to be dropped, and then I moved on. Some of the challenges, just to see, the Plasmodium falciparum genome is AT rich. It's like 80% AT. So when you try to translate this genome in E. coli or in Pichia pastoris or in CHO cells, there is premature termination because our genome is not that AT rich. So to avoid that, we utilize something called codon harmonization. And this basically, it changes the codons, and we were able to map out those cysteines which were involved in making these disulfide bonds, and we were able to insert rare codons there. So the idea was that when the amplicon goes there during the translation, it take a pause, and that pause will give it some time to make that particular disulfide bond. And it was really difficult because of the conformational epitopes, as I said. They have to, certain disulfide bond has to form in a certain way to make it functional. And culture and maintenance of infectious gametocytes, it was another. They have to be changed every day. The blood has to be changed every day when you want to maintain them in the lab. And they are temperature sensitive. If you drop the temperature less than 37 degrees, they're gonna be non-infectious. They're gonna fertilize in vitro, basically. They will not wait to be ingested by the mosquitoes. And for Vivax, you can't even culture them in the lab. So one of the vaccine candidate I was working, it was for Plasmodium Vivax. For that, we have to collaborate somebody in the Thailand. So when some person came with the malaria and they figure out that it's Vivax, they will take the blood from that patient and they will feed it to mosquitoes. So Plasmodium Vivax can't even be cultured in the lab. So that's why it was very, very difficult to make the vaccines against malaria. But we took our chance, we tried it, and it didn't work when we scaled it up. It was perfect in the lab, but when the technology went to the private partner to manufacture it under good manufacturing conditions, GMP, good manufacturing practices, it did not work. So while I was working with this particular vaccine and we were trying it in research macaques, I came to know a group at Tulane Primate Center, and it was quite a unique place. I think US has only 6 where you can work on monkeys. The place is highly regulated, and it's extremely expensive to maintain monkey colonies. And I think somebody told me that it takes like $100,000 per year to maintain a monkey. They are veterinarians, so it's more expensive to maintain a monkey than a human being. So there were very tough regulations, but I was able to get a chance to work there, and it was really, really amazing. So we worked on HIV, SIV enteropathy, and monkeys are not, they don't get infected by HIV. So they the model is using SIV, simian immunodeficient virus. So we were able to create hybrid viruses. We used to call them SHIV, SHIV. So we'll take a portion of HIV and put in SIV. So it can infect the monkey, and we can study the biology of it, the pathology. So when we talk about enteropathy, the GI tract, the gastrointestinal tract, it's a major target of HIV, SIV infection. And the damage to mucosal immune system is associated with a variety of gastrointestinal manifestation, collectively called as AIDS enteropathy. Generally characterized by chronic diarrhea and wasting. And nowadays, very good antiretroviral drugs are available. So the patient who have HIV, they are living for very long times, and they always have trouble with diarrhea. So their quality of life is really, really bad. So it's linked with immune activation, inflammation, and viral replication. And I will get back to that topic in the next slide. And we also were looking at epigenetic mechanisms. So we were looking at microRNAs. Can they post-transcriptionally regulate cell proliferation, cytokine signaling, and inflammatory response? So as you can see in this slide, so in a perfectly healthy person, that's the case. There is colon, there is your gut, and that's the perfect balance. But when you have HIV, the virus, there are tight junction proteins, and they lose their function. So there is a passage. So all the harmful bacteria from colon can enter, and they can cause immune activation. And immune activation is very good for the virus. If the cells are activated, virus just goes in and it replicates. So there is high replication. So what we were trying to do is we were trying to get the effect of THC, that's the active component of marijuana, and try to see its anti-inflammatory effect in monkeys, and we got some very good results. So I think the people who are living with HIV for very long times, marijuana can really help them to bring down the inflammation in their gut, and they can enjoy a better quality of life. So these was a couple of the questions we were asking there. So identify.
Is it legal to allowed marijuana medical use?
Yes, it is getting there. I mean, a lot of states already are legalizing it, but we were hoping it more like for example, like a injection or something.
Okay, medical purpose.
Medical purposes, exactly. It's very good for anti-inflammation, and almost 80% of the disease, they come from inflammation, because the kind of food we eat, the kind of environment we live in, is always there is continuous inflammation in the gut. So that's what basically turned out to be the causal reason of a lot of diseases. So I think that can really help. And marijuana has been used in Indian culture in different ways. I mean, it was not that demonized. So I think they understood the therapeutic potential. So once used under the guidance of a medical practitioner, it could really be helpful. So we were trying to do the microRNA profiling in the intestine associated with the immune cell activation, viral replication. We just want to see if we can close these leaky gut syndrome, if we can close these tight junction proteins, and the bad bacteria cannot cross the epithelial barrier. And what are the epigenetic changes when that happens? We want to identify the genes regulated by the immune activation associated with microRNA in the intestine. And are there any differential expressed microRNA targeting SIV? That was a very good question. And then effect of THC on intestinal anti-inflammatory microRNA response. So the work there came out to be very fruitful, and we were able to publish some pretty good work. So these are some selected publications, a couple of in Journal of Immunology, a couple of in Journal of Virology, Frontiers in Immunology. So the stay at Student Primate Center was very, very fruitful for me. But then I realized I was really sad about the transmission blocking vaccine story, because it just kind of, the project was killed. It could not be manufactured in the private lab in the industry, and that was it. We get a couple of grants, we write a couple of manuscripts, but that's about it. And I was always interested in applied sciences, how we can utilize this knowledge, this expertise in making actual drugs, which has an actual chance to go to clinic and help patients. And the funding situation in US was not very good as well at that time. So that was one of the reasons too. I don't think I'm going to sit down and write grants day and night. That was not the reason I went in the science anyway. I was always fascinated by the applied part of it, how it could be applicable to help patients or for diagnosis and whatnot. That has been driving force for me to choose this career. So I joined the pharma business basically, and this slide shows you how it works. So very first phase is discovery. So in discovery, you try to choose the leading candidates. You have certain candidates, you characterize them, and you find the best one to go forward. Then you go to the preclinical. Preclinical is the animal studies. Once you have the molecule selected, you inoculate them in, FDA asks you to do it in at least 2 animal species. So either monkey and mouse, rat or dogs, depending on the relevance. You have to do it in 2 animal species. And after that, once you have those results, if they look good, then you go to phase 1, which is basically safety. You put them in human phase 1 trials. Phase 2, safety and efficacy. Phase 3, again, then registration, and then final, the product launch. But as soon as you put them in animals, you have to do these testings. Immunogenicity testing is the first, because it's about safety, what it does to the animals and later on human being. Are there any unwanted effects? And if there are, how serious they are, the likelihood of it and the seriousness of it. It's like a risk-based approach. What is the risk? What is the probability is going to happen? And how bad it can be. So those were the 2 parameters you always keep in mind when you go for the safety assessment. biomarker analysis, and of course, pharmacokinetics. So as you can see, it starts from preclinical as soon as you put it in the animals, and then it will go all the way till launch. And now FDA even asks post-marketing surveillance, because when these assays were being developed, and the technology is changing every day. So we have more and more sensitive technologies, and we can have more and more sensitive assays. So FDA asks you to do the post-marketing surveillance as well. So if something happens, it has to be sent back to the labs, and they have to see really what's going on. And these all tests are required by the FDA, by the EMA, if you want to launch your product. So they have to be done. And they have to be done in a regulatory environment, GLP facility, good laboratory practices. They cannot be done in our academic labs. And they have a different way of doing it. And I'll go through them, some of those points, how it is different than our usual research labs. So what is immunogenicity? So immunogenicity is basically intrinsic capacity of a product to induce an immune response in target population. And it could be mainly of two kinds. One is wanted and one is unwanted. And wanted is the case when you use a vaccine or immunotherapy. You want it to produce antibodies. You want it to give you the safety. So you want it to be immunogenic, and that's wanted immunogenicity. However, the biologics these days, especially the protein therapeutics, they are designed to exert a specific therapeutic function. It's a biological activity. It doesn't necessarily want it to raise any antibody. So it is not designed to basically raise any immune response, but it does sometimes. There are various factors, patient-based factors, the nature of the protein itself, and the condition of the patient, if they are immunocompromised. So there are these anti-drug antibodies, and they could have very serious safety consequences. And I'll go through them with some examples in my presentation later. But they can really reduce efficacy, and they can compromise the PK/PD and safety, and can have very serious side effects, which can end up in canceling the product, taking it out of the market. And there have been several examples of that. So a lot of careful planning and a lot of careful assessment has to be done. And FDA and EMA, all the regulatory agencies require it at the time of filing, when you file it. So the immunogenicity can manifest in various ways. It can be T-cell activation, hypersensitivity, anti-drug antibodies, innate immunity. But for this presentation, we're going to focus on anti-drug antibodies and the T-cell activation. So the factors, what is it? So first thing is the protein. Which species this protein comes from, which epitopes it has, which kind of post-translational modification it has, which kind of glycosylation profile this protein has, which kind of expression system you are using to make it, the product, the host cell proteins. And it becomes really troublesome, because if you make these biologics in, let us say, CHO cells, in eukaryotic cells, some of the eukaryotic cells proteins are co-purified with the product. And how they're going to affect the immune, they are supposed to be highly immunogenic. So there is a lot of wide testing areas. They have to be really, really minimal. And the trouble with host cell proteins are that there is no known minimum quantity. So you cannot say, oh, there are very few, it's not a problem, because there is no minimum concentration which can increase the risk of immunogenicity. So you have to be really careful to eliminate them and to get as low as possible. The second is aggregation. If your therapeutics is aggregated, it's going to create an immune response. The formulation, which kind of formulation you are using. So all these factors from the product and from the protein. And then the second is clinical factors in the patients. Does the patient have immunocompromised status? In case of cancer patients, I mean, their immune system is almost devastated by that point due to all the chemotherapy and all the other drugs they take. So they are very prone to make ADAs. And they can have serious troubles. Their immune system cannot handle it. The treatment, the route of an administration, are we giving it subcutaneously? Are we giving it IV? IV is the safest route. It has the least probability to start immunogenicity. The dose, is it a single dose agent, or you're going to do it multiple times? If it is a single dose, it's very easy. There is very less chance of making anti-drug antibodies, the ADAs. But if you have a repeated doses, then it increases the chance significantly. And all the risk has to be evaluated very carefully. So that's what basically I do in safety assessment department. That's the overall scope of immunogenicity assessment. So once you have a compound, a leading candidate, you see in the published literature what is the probability and what is the potential impact. What are the therapeutic properties? Which kind of patient population we are targeting? So from the product point of view and from the patient point of view. So you get all these things tabularized, and this document has to go to FDA when you ask them for initiation of a new drug. So you have to do the assessment before you start anything, whatever information is there on the public domain. So you have to do a thorough literature research, all the animal studies which are available, and if some other company has used it in humans, what was their data? So a comprehensive review. Sample collection and timing. So when you are in the clinical trial, in animal studies or in human studies, what is the best time to take the sample? Because the ideal time would be when there is no drug in your system. Because if there is drug present at the time of taking the sample, it can interfere with your immunogenicity assessment. And I will touch on that a little bit later in the presentation. The kinetics of isotope development, related to the serum drug levels, less serum drug levels in the serum is better for the assessment, is better, less interference in your assay. But these maps these days, they stay long, very long. So no matter you do the sampling after 21 days or 31 days, you will always find them there. So when you design a ADA assay to assess the immunogenicity, you have to really think about it. You should know the mode of action of the drug and how long it stays in your system. And if it is there after your first cycle or second cycle, how are you gonna solve that trouble? Which kind of approach you will take when you design an assay so that there is no drug interference and the immunogenicity data what you are producing is reliable. Detecting ADA responses, that will be the main focus of this talk. So it's gonna be designing of these assay, setting cut points. So the trouble with ADA assays, there is no positive control. For example, for PK, for the pharmacokinetics assay, for biomarkers, there are standards. So you have a standard dilution, you get the standard curve, and you know the quantity in unknown. It's very easy. But for ADA, there are no positive controls. And since we cannot hyperimmunize a human, we always have to do with this, what they say, surrogate positive control. So you have to raise the positive control in animals, in rabbit, in sheep, and that's where the trouble starts. So what's happening in a sheep does not exactly tell you what's happening in a human. And even in a human, we are outbred mammals. So everybody has their own unique genetic composition. So every person is different. Every person will react to this particular biologics in a certain way. But when you use animal positive control, it even complicates things further. But that's the best we can do right now. So that's what we have to do. So you have to set a cut point basically. So I'll go in the little bit in details how we do it. But it's really interesting how everything is relative in ADAs. When you detect the ADA, it's relative to the population you use to set up your cut points. There is no hard and fast rules. And you have to be very careful, because if you are using different population than your target population, which you are using your drugs in, there could be huge interference, there could be huge difference in cut points. You can have a lot of false positive or false negative, and that can throw off your whole study. So it has to be really designed and checked very carefully from the beginning. Once we have the ADA response, then we have to characterize it, the neutralizing assay. You have the ADAs, these anti-drug antibodies. What do they do? Sometimes they don't do anything, which is good for everybody. Sometimes they make neutralizing antibodies, which basically neutralize the effect of your drug. And if it cross-reacts with the endogenous part of the drug, it can become very serious safety event. So we have to, after you have the ADA, then you have to develop a neutralizing assay as well to make sure that the patients who have these anti-drug antibodies, they are not neutralizing. And if they are neutralizing, what actually they are doing? How serious is it? The isotyping of it, the affinity, and the epitope mapping. And finally, the reporting ADA response. Every company has a different way to report the outcome of clinical trials. So it's basically patient-based. There are two main categories. One is treatment-induced. So all the patients which are enrolled in a clinical trial, they are taken a baseline level of ADAs. They take a blood sample before they start the treatment and they analyze that. And then once the treatment starts, after every cycle, 21-day cycle, 31-day, whatever the case may be, there are blood drawn and it's continuously monitored during the whole study period, the whole trial. So if a person has no antibodies before he start the treatment, and after taking the drug, he start developing ADAs, he is categorized as treatment induced antibodies, treatment induced ADAs, since the person was negative before and became positive afterwards. If a person has some low level of antibodies before it started the treatment because it was taking some other drug which was homologous, or there was some other environment, let us say in case of PEG, if you are eating ice cream or if you're using certain kind of shampoos, so if you're kind of exposed to that particular product and you have the pre-existing antibodies, but after starting the treatment, the titer goes up 4 times. So that's called as treatment boosted. So there are several categories. In some patients, it's going to be persistent. The cycle 1, once they are ADR, they cycle 2, cycle 3, cycle 4. So they are called persistent positive, and they have to be followed very carefully because they might have trouble in the efficacy and safety of the product. In some patients, it starts at cycle 3 and then it goes away. So if there is no consistent ADA level in a patient for 16 weeks apart, so sample 1 was taken at 16 and sample 2 is after 16 weeks, and if it's not consistent, then those patients are called as non-persistent patients, and they are kind of low in the risk profile. So a lot of careful planning, a lot of careful interpretation has to be done before you submit this reporting to the FDA, because it's make or break case. If it's a safety profile is not good, they will not approve it, and all the money you utilize, the millions of dollars, is basically down the drain. It's in waste. So it's extremely important. You have to be very careful, very thorough and it's kind of interesting. Everybody is from PK, PD, they all reach out to me. Hey, what's going on? So nobody can move further until you give them a green light. So safety assessment, this is very, very important, especially when you are developing new therapeutics. So extremely exciting. Traditional antibody response. What happens when you give a new therapeutic to the patients? So after first inoculation, first immunization, is generally IgM, as you can see in the blue. But later after the second and third, as the weeks go, after 4th week, the IgG start coming in. So IgM is not that troublesome. It's generally transient, it goes away. But IgG could be real troublesome because all the reported neutralizing antibodies, they are IgG, they're class switching IgG. So as I was saying before, if you have only one dose, then it's not that big a deal, because if you will have immune response is going to be IgM. But if you have to give the dose repeatedly, you have to be very careful in sampling, and you have to really see what's going on after every cycle. So the primary response is IgM. It follows a lag phase, it generates memory, and it declines to near the baseline after some time. So it's not a big deal. But when the secondary response, when you do it repeatedly, when you give the repeated dose, it produces higher titers, stronger affinities, predominantly IgG, and there is always a possibility of neutralizing antibodies, NAB development. There is isotype switching, and they sustain. So they are there. So after the treatment is finished in a clinical trial, and when the patient go in safety follow-ups, we still collect blood samples from them just to know if somebody has some antibodies, what they're doing. Are they harming him after the study is finished? So safety starts from very beginning, before you start, all the way to patient follow-ups, and in some cases even after the drug is marketed, even then it is being followed. The characteristics of anti-drug antibodies, ADAs, they could be binding, and they are the best kind and most common. They just bound to the drug, they don't do anything, there is no apparent effect, they are just there. Sustaining, The ADA drug complex, it prolongs the circulating half-life, so it increases the drug exposure, and you have to see what's going on exactly, how it is translating in the safety profile. Is it creating trouble? Are you getting better efficacy, or are you getting more toxicity? Clearing, it can decrease the drug exposure. The ADA drug complex, because of the size, it will be seen as foreign particles and they will be cleared up from the system. So you can have trouble with efficacy in that case. Or if it is neutralizing, that's the worst kind of thing, because it can decrease efficacy and it can have very serious safety events. The good part about clinical immunogenicity is that the severity of ADA impact and its frequency has been inversely correlated. So the high frequency is for binding ADAs which don't do anything, we just are there. And the very high impact is the cross-reacting neutralizing antibodies, but their frequency is very low. So that's one thing is in favor of us. These are some of the examples, known examples: loss of efficacy because of ADAs in insulin, streptokinase, factor VIII. It's very very known case. Interferon alpha 2, interleukin 2. So that's what we did in Nectar. Interleukin 2 lost efficacy because it was highly reactive, but we were able to pegylated it and made it more safe with a better PK profile, with better safety profile. And now we are trying it in at least 20 indication of different kind of cancers, alone as a monotherapy and in combination with some of the pre-marketed product by Bristol Myers Squibb. Enhancement of efficacy, growth hormone. So it can be trouble as well. If it is overworking, then you're going to have troubles with it. Neutralizing, erythropoietin EPO. This is a very textbook example. So what happened is that it was erythropoietin. So when they give the EPO to people, it made neutralizing antibodies, and those neutralizing antibodies cross-reacted with endogenous erythropoietin, and basically it wiped out erythropoietin from the people. So these patients ended up having severe case of anemia, and the situation was really life-threatening. So they had to take it out of the shelf. So we have to be really careful when we kind of characterize these neutralizing responses, because they can really put you down. Generally, allergy, anaphylaxis, and serum sickness, these are the very common minimal one. So as you can see, the range of the effects can go from simple allergy all the way to severe anemia, life-threatening condition. So a little bit, we're going to go now in the prediction, what we can do before we put it in the animals or humans. There is a lot of planning and analysis goes in silico analysis. So immune response to therapeutic proteins is characterized by the generation of antibodies, and they can be T-cell dependent or T-cell independent. T-cell independent antibody response generally result in low affinity IgM antibodies, and it clears out after some time. So it's not a big deal. But T-cell driven IgG ADA can be really, really dangerous, and they have a great potential to react with endogenous counterpart, and they can class switch and become neutralizing antibodies. So whenever T-cell is activated, it's a bad news. So we do everything in our power to predict the T-cell epitopes, and if there are some epitopes, can they be modified? Can they de-immunize it by changing the sequence, just to avoid the serious consequences down the road? So as this, there are several companies which give these services. We happen to like Epivax, and they have very good platform, and they provide very good insight as far as the immunogenicity T-cell epitope prediction is concerned. They have very good proven algorithm, which is being used by Pfizer, Eli Lilly, all the big boys. So we use them as well. So if T helper cell is activated, because there are T-cell epitopes in your therapeutic protein, then it triggers the T-cell dependent antibody response, which is mainly IgG, and which can turn into neutralizing antibodies and can do serious damage to your product and to your body. If there is no T-cell activation, then there is no B-cell activation. there is no AD response, whatever happens is generally IgM, which could be cleared by itself. It's not a big deal. So we try to make predictions. What does the antigen presenting cell present? And what does the T cell see? And how does it affect the immune response? So T cell epitope and immunogenicity analysis, they go hand in hand. And the good thing about T cell epitopes is they are linear. So there are very good algorithms available which can detect them very efficiently. With B cell, since the epitopes are conformational, it's not that straightforward. So the prediction software doesn't work as good for B cell epitopes as for the T cell. And we can do a lot of things by predicting these T cell epitopes in the therapeutic protein sequence. We can, of course, do epitope mapping. We can do comprehensive assessment of immunogenic potential. If certain T cell epitopes are conserved in the population, what did they do with other products? And you can get a better understanding if that particular sequence is present, if that particular epitope is present in my candidate, what can happen, how it can manifest down the road. Vaccine design. Opposite to biologics, in vaccine, you want to have immunogenicity. So you can design a better antigen, which will give you robust immune response, which make robust antibodies once it's in, and which help you protect from the disease. The protein deimmunization. If your biologic protein sequence has a lot of T cell epitopes, some of the amino acids could be modified, and it will result in low immunogenicity score. You will have a better chance to succeed. And of course, immunomodulation. So there are 3 different kind of tools. First is in silico, in which they screen multiple therapeutic candidates, then rank for immunogenicity based on predicted epitope concepts, the epitope content, and then they modify the sequence, de-immunize it to reduce the immunogenicity. So that's all done in silico with your protein sequence. Then they go in vitro and ex vivo. They do the HLA, human leukocyte antigen binding studies, cell-based assays, memory and T cells, the whole protein screening. And then they have some in vivo assays as well, in which they have human SCID mouse model and HLA transgenic mouse model. So they can see how tightly it is binding with HLAs. Preclinical immunogenicity. Preclinical is when you basically inject your vaccine or therapeutic candidate biologics in animal species. And the downside of it, it doesn't predict what it's going to do when you're going to inject it in the humans. So why to do it? Because if you have weird PK results, if your pharmacokinetic results are not adding up, you see sudden clearance, ADA could be one reason, because since there are ADAs, that's why either the clearance is faster, the therapeutic is getting out of the system very fast, or it's being delayed and there is toxicity. So it's only good to interpret the PK results as far as the preclinical animal studies are concerned. Then why we do it? Because it's very important to know as much as about your product as early as possible. As you can see here in the IND, preclinical lead, phase, and phase 2. This is called value of death. Only 0.1% preclinical candidates make it to commercial market. So that's 1 in 1,000. And these phases are extremely expensive, multi-million-dollar investment. So you want to make sure that you know everything you can before you start the lead candidate. So that's why you use all the in silico tools, all the preclinical tools to know the immunogenicity profile and the product and process related risk, stability, aggregation, which can create trouble down the road. So that's why we do the preclinical immunogenicity assessment, and it's very important. So good thing about T cell immunogenicity is that the T cell epitopes are linear. So for class 1 human leukocyte antigen cell surface receptor HLA, they are 9 to 10 amino acid in length, and for class 2 they are 9 amino acid. So what they do is they take the protein sequence and they change it in 9 modes. So 1 to 9 is. And then they have something called epimetrics. Epimetrics is a matrix-based prediction algorithm. And if they have it, they can do a blast search and find out some of the conserved regions, some of the conserved epitopes which has been troublesome, and they can give it a immunogenicity score. They can also predict the MHC binding, which is prerequisite for the immunogenicity. So that's how they give the score, the presence of certain sequences, certain epitopes, and how tightly it bind with MHC. So let us say if you have 10 candidates, you can find out which one is gonna be least immunogenic before you even start it. So that's how they try to do. It's a simplified. So they scan the whole protein sequence. They get the epitopes. So there is 3 epitopes, 1 plus 1 plus 1. If some other sequence has 2, that means it's less immunogenic. It looks pretty simple. But they even went further. There are 2 kinds of T cell epitopes. One is the T cell effector, and the second is T cell Tregs, T cell regulator. So the effector epitopes turn on the immune response, and the regulator shuts it down. So they have very sophisticated algorithms in which they can find out, they can distinguish between Treg and T effector epitopes. So they can have much better, much accurate prediction of the immunogenicity. So for example, here, there are 2 T effector epitope and 1 is T regulatory, Treg. So they're gonna add up all the T effector and then they minus the Treg because it's gonna put the immune response down, and plus HLA of the subjects. So by utilizing these sophisticated algorithms, you can get a much better picture of the immunogenicity prediction for your candidates when you are selecting which one you want to go forward with. Extremely effective and very cheap, if you say, because everything is done in silico. There is no money involved that much in clinical trials or in animal studies yet. So everybody has to do it early in discovery phase. Now the FDA expectation. So these are the guidelines which were the latest one, which came in 2019. Immunogenicity testing of therapeutic protein products, developing and validating assays for anti-drug antibodies detection, guidance for industries. And it clearly says that the sponsor should provide an immunogenicity risk assessment as well as a rationale for immunogenicity testing paradigm in the original IND. So whenever you start any trial, you have to start developing and validating assays to assess the immunogenicity, to see if there are ADA generation. If there are, then you have to characterize them. Are they neutralizing? If they are neutralizing, what do they actually do? So as soon as you start a new drug application, you have to have all the plan in process. And during the time of filing, when you file the data, you have to provide an integrated immunogenicity summary, which will summarize all the results you get from all your clinical trials. Without that, it's not going to go forward. It's not going to go anywhere. So it's extremely important, and everybody has to do it. These are some of the guidance documents. EMA, that's European Medicine Agency. So if you basically, the drugs are developed for the global market. So in the USA, it's the FDA, Food and Drug Administration. In Europe, it's EMA, and then every country has their own regulatory agency. But everybody more or less follow the same guidelines as EMA and FDA with a little bit of variation. So all these documents say that the approach to do the immunogenicity assessment should be risk-based. A risk based on likelihood of the immune response, how probable is it, and the consequence, how serious is it, and which kind of approach to be taken for immunogenicity testing, type of testing, and timing of the testing. So these all approaches are risk-based. How probable is it to happen, and how bad it can get. So now I'll finally start about something about ADA assays So there are two kinds of assays. Ones in which the reference standards are available, and they're pretty straightforward. And it's generally PK assays, the pharmacokinetic assay, the biomarker assays, and the potency. So they are definitive and they're quantitative. You can exactly know how much drug is available in that particular sample. In case of ADA, they are quasi-quantitative. And what does it mean? They are not quantitative. You cannot tell how many milligrams or nanograms of antibodies one particular sample has, because there is no standard curve. And real trouble happens because, as I was saying before, that the positive control is raised in animals. So there is difference of species. What's happening in the rabbit or what's happening in the sheep does not really reflect what's happening in a human being, but that's the best we can do. So we have to do it. As you do the dosing, repeated dosing, the maturation of immune response and the composition change. And the results explained in terms of characteristic of the test are like in tiers. So FDA say there should be 3 tiers. Precision but not accuracy can be demonstrated. So these are certain limitations because these are not definitive quantitative assays. They are relative. So when you set a cut point, and cut point is the point above which a sample is considered positive and below which is negative, a lot of careful consideration about which samples you're going to use and which kind of criteria you're going to use for false positive. That can change everything. And I will come to that later in the presentation. So composition of anti-ADA response, it's single or multiple epitope specificity, isotype mixture. So there are going to be variations between people to people because everybody has their own genetic makeup, their own immune system. But even a same person, once you do a dosing repeatedly, every time the isotope changes. So it's very highly variable. So in practice, the exact and unique composition of each sample is not known, but we should do the best we can with the techniques available, and it has to be done. There is no way around it. So it's very important to understand that, you know, whatever comes out of the immunogenicity assessment, that might not be the ultimate truth because it's relative, it's not absolute. As I was saying, the positive control, it has to be raised in animals. Polyclonal is preferred than the monoclonal. But if the biology you are developing, if you have to be, it has to be given like continuously, multiple times, then the polyclonal as well as monoclonal positive controls are used. So you have to show that during the validation that the monoclonal positive control is as good as polyclonal. And some of these studies go for 10, 15, 20 years. So let us say if you have a goat or a rabbit, it has to be maintained. It has to live well, and you have to pay a lot of money for the maintenance so you can get positive control time to time from it. You can bleed it and you can get the serum. However, in monoclonal antibodies, you can just put them in -80. So if you can show to FDA in the assay validation that positive control, which are polyclonal and monoclonal, they're kind of comparable, then you can get away with it. And maintaining monoclonal antibodies is much more easier over a long period of time than the whole animal. So there are certain things. The choice of assay format, of course, may influence the collection and the selection of species. Immunization more than one animal is accounted for non-respondable. You have to have multiple animals and they have to live really long and happily. Something happened to them, it could pose a serious problem because something happened 2 years down the road and you don't have enough positive control. And there are thousands and thousands of clinical samples from the clinical trials. How are you going to analyze it? So that's why it's very important to plan these things in advance properly and try to make polyclonal and monoclonal positive control at the same time. What about the negative control? So we use commercial vendors and they send us the sera, normal human serum, and that's being tested. Some of the sera has a lot of inhibition. They might have something, they might have been exposed to something. So they all are screened out. And it has to be really mixed as far as gender, age and geographical distribution. And it's best to match your target population. If you are doing a clinical trial in Africa, it's better to have these samples from that population, because they live in a particular environment, they eat a particular diet, they're exposed to particular things. So people living in the US might not have the same repertoire as the people who are living in Africa, because they're exposed to different things. So it's very important to choose these controls, especially the negative control sera, from the same population which you are conducting your clinical trial in. Now which format you're going to do. So, and ADSA format can have huge implications as far as the results are concerned. ELISA is the favorite, but it results in higher MRD and lower sensitivity. Drug tolerance is the problem. And if you are not careful about the drug tolerance of your assay, the reported immunogenicity may be highly underrated. And it has shown in this publication. So they reviewed 28 FDA products, and they realized that there was very high concentration of drug in the patients when the samples were taken and when they were assayed. And in case of one interesting case is Humira. It's a very popular drug, popular mAb, monoclonal antibody. So the drug tolerance of the ADSA was 2 microgram per ml, and the presence of drug in the humans, in the samples, were like 8. So it was 1/4. So all the immunogenicity which was reported, it was kind of bogus. It didn't make any sense. So FDA asked them to do the post-marketing surveillance. So when the better assay, better technique were available, they have to go back and analyze those sample again and show the immunogenicity data. So I'll go quickly about a couple of formats. Is the favorite format, the bridging assay, they say. So there is a streptavidin plate. You biotinate your drug. It binds to the streptavidin. So your drug is kind of immobilized on the plate. You apply your sample, and then you add the ruthenilated drug, sulfotag, which is this is your capture, and this is your detector. So the antibody ADAs, which are present in the patient serum, they make kind of bridge between them. And once they make the bridge, they give a signal, and you read it on MSD, mesoscale discovery. It's very, very, very sensitive. Generally, we use ELISA readers in our research labs, but ELISA doesn't give you that kind of sensitivity, nanograms. So we use MSD in industry. It's high throughput, very specific, and it's possible to use any positive control. It has very low backgrounds. The disadvantage is the sensitivity, restricted detection of low affinity antibodies. It only favors the high affinity. And once you modify the drug, you biotinate it or you ruthylate it, it can denature epitopes. It can cover them. So it can have trouble in detecting the ADAs. And it's susceptible to interference by therapeutics and serum component. In that case, for example, for us, we use pegylated drugs. So we use direct ELISA. So you coat the drug on, you apply the sample, the ADAs are bind to it, and then you use the detector molecule, detector antibodies, and you read them. It's ready to do in HRP, or you can do it in MSD format as well. So very sensitive, very reproducible, very robust. Easy to use and automate, comparatively inexpensive. There are commercially available secondary antibodies, so you don't have to raise your own. The downside is that source of positive control has to be the same as ADA, and the specificity is a trouble. Sometimes it's not that specific. Again, detection of low affinity antibodies is a problem, and sometimes it can end up in high background. So you have to very carefully, during the development of assay, you have to be very careful to address all these issues. When we talk about ADSAs and regulatory expectations, these are the 3 cornerstones: the sensitivity, the drug tolerance, and the target interference. And assay has to be highly sensitive. And that means is that it should be able to detect very low amount of ADAs. And back in 2013, I think, FDA wanted like 250 to 500 nanogram per ml sensitivity, but then they changed in 2016. And now they want 100 nanogram per ml. at least. Our methods which we develop, which we use, they give almost 10 nanogram per ml sensitivity. And as I was saying before, your assay has to be drug tolerant. If drug is present in the sample when you are analyzing them, it should not interfere. Without drug tolerance, the sensitivity doesn't mean anything. And as I say, in case of Humira, when there was 4 times more drug present than the drug tolerance of your assay, it doesn't mean anything. You have to very carefully look in the target interference, because it can end up in false negative and false positive. So these three particular, the selectivity, the sensitivity, target interference, and drug tolerance, they have to be kind of balanced, and a lot of careful thought process and a lot of developmental work should go when you start developing an assay to get a robust assay and to get reliable results. So Fda expectation for sensitivity is 100 nanogram per ml, and is determined using positive control antibody, and you need affinity purified antibodies. The minimum required dilution, MRD, if you dilute your sample too much, you can have trouble. So generally you use 1:25, 1:50 in that range to get the balance between sensitivity and selectivity. And you have to optimize your concentration of labeled reagents. That's what we do when we do the assay development. Use different concentration of coating antibody, coating material, your capture, your detection. Drug tolerance is another very important one. Presence of free drug in sample may interfere with the detection of ADA. So you should really know how much drug it can. During the development, you have to find out how much drug this assay can handle, and it can still give you reliable readings. If there is a lot of drug available in the sample, which happens with the monoclonal antibodies, because they hang around for a very long time in the system once you take them. So what you do is you basically buttonlate your drug and you capture it on streptavidin plate, and then you acid dissociate the antibody and drug complex. You use acid at low pH at around 3, they dissociate. After that, you neutralize it, and then you let them bind. So once these ADAs bind to these immobilized drug, then you do second acid dissociation, you dissociate them again, and then you finally assay them. So what happens is you get rid of all the free drugs which is hanging around the sample, which is creating interference. So you capture them on immobilized drug, you dissociate them, and then you finally assay them. So there are various tricks to go around the drug tolerance problem. And it was really huge in earlier days, 10 years back, it was a huge trouble, but now they are getting better results. They have more techniques, there are technological advances. So these 3 things, FDA look very carefully when you show them your assay design and how you are assessing the immunogenicity. Target interference, it can occur when the target is soluble, so it's already there in your sample, and it's multimeric, like IgE and angiopoietin, and it can give you false positive results, which is a real big trouble. So ADSA, when you develop, you should have mechanism of action for the molecule, so you can see what is it doing. And this is that's the graphic representation of a bridging assay, in which the ADA present in the sample, patient sample, give you the bridging and give you the signal. But look at here, that if the target is multivalent and it's freely, it can do the same thing. So this is positive and this is false positive. So you have to, it's a big trouble in rheumatoid arthritis, Rh factor. It's a huge interfering, there is a lot of target interference in those populations, and you can have a lot of false positives. So you have to be very clever, and you have to decide which format to follow. Fulranumab is a very good example. It's a neutralizing nerve growth factor. And when they did the analysis, they were like 70% samples were positive, which was not possible. So they revised their ADA assay. They first removed the nerve growth factor using anti-NGF antibody-coated beads, and then followed by competitive inhibition with fulranumab. Of course, more than 50% ADA samples which were positive, only 1.3% was confirmed. So you have to really pay a lot of attention which population you are using, and if there is any interfering substances. in that particular population, or if you have the target interference, it has to be addressed during the assay development and assay validation to avoid false positive or false negative results. That's the immunogenicity workflow. All the samples first go to screening assay, then all the positive from there to confirmation. Then after that, we do the titration. And after that, we do the neutralizing antibody assay. I think I'm kind of running close to it, so I will go really fast. If you have any question, please let me know. So this is a 3-tier testing strategy devised by FDA. You're gonna screen all the samples, and during screening, you have a 5% false positive rate. So you are kind of making this big possibility to catch all the binding antibodies, no matter they're specific or not. If they the sample has antibody, they should be detected, and it's okay to have false positive. After all the false positive with 5% expected rate, all the samples which are screen positive, they go to second tier, and that's confirmation, and that's being done in the presence of drug. So the binding is specific, and you want to eliminate any false positives. So you have to have 99% confidence. So there is only 1% chance of false positive. And finally, you characterize the titer. How much is kind of amount of response roughly is not really nanogram per ml, but you just give a sense how much antibody this particular sample has, which isotype, and is it neutralizing. And to do this, you always use the cut point, assay cut point, and it's determined by the data from drug naive patients statistically. And a sample is considered putative positive if the response is greater than or equal to the assay cut point. So that's the main trouble. You have to be really careful which population you use to set those cut points, because there is no standard curve. So tier 1 screening assay, 5% false positive rate. If it is above cut point, it's called putative positive. It go to tier 2 in confirmatory assay. If it's again the response is below cut point, it's negative, and we report it as an ADA negative. The patient doesn't have it. If it is above cut point, it's called positive. Then it go to tier 3, in which we determine the titer, how much antibody it has roughly, and characterize the response. Is it neutralizing? What is the affinity? What is the epitope? And what is the isotype? So that's very standard 3-tier sample testing guided by the FDA, given by the FDA. The ideal assay, when you look for it, it should be sensitive enough to detect very low level. It should detect all isotypes. It should have no false positive. And since it's a screening one, it should have 5% false positive. And should discriminate between pre-existing antibody and treatment induced. And I was telling that if the patient has antibodies before it start the treatment or during the treatment, it should have good drug tolerance, and it should be reproducible. So these are the main points we do when we validate an assay. And the question comes, why to validate an assay anyway? We run the assays in our research labs all the time. Why to do it? And the reason behind it, these studies go generally very long, the clinical trials. They go for like 10 years, sometimes 15 years. They go in multiple locations in the world, in China, Africa, Asia, Europe. So multiple labs do the same assays. So how to make sure that the data is being produced is comparable? Because there is day-to-day variability, plate-to-plate variability, analyst-to-analyst variability, lab-to-lab variability. So you need a really robust assay. And so when the data is produced in Europe, in US, in China, wherever, the data is reproducible and is comparable. So that's why you have to validate the assay. These are the main thing we do: screening cut point, then confirmatory cut point, precision, sensitivity, matrix interference, drug tolerance, robustness, and stability. So very quickly, I'll go to the cut point determination. Generally, we use 50 drug naive subjects, the people who don't have the drug yet. And these samples are run in 6 runs by 2 analysts, 3 runs per analyst. Each sample is tested in duplicate. That's how it look like. So at least 2 analysts, and they're gonna run 18 plates. And the reason behind it, all the sample get tested in every run and on every plate at different spaces in the plate. So you rule out day-to-day variability, you rule out analyst-to-analyst variability, and you rule out the variability of the sample in the plate itself, because sometimes the beginning of the plate is hard, sometimes the middle, and sometimes the end. So that's how it actually look like. So you do the negative control, and you do all the samples with drugs as well. In the presence of drug, to get confirmatory cut point. In the absence of drug, you do it for screening cut point. So that's how it look like, all the samples with drug and without drug. Once you have the data from these at least 18 to 20 plates, it's a lot of data. You remove high CV samples if the same duplicates, one has high value than others, coefficient of variation. You normalize and transform the data. You remove outliers, analytical and biological, and then you calculate the cut point. And then you decide if the data is parametric or non-parametric. So that's what you do for a screening cut point. You get this data from 50 drug naive subject, 6 total runs by 2 analysts. You normalize the data. You identify and exclude the outliers, and then you evaluate the screening cut point factor. If you do a Shapiro-Wilk test, the p-value is less than 0.05 and the data skewness is more than 1, then you go with non-parametric 95 percentile. That means 5% false positive rate. If the data is normally distributed, you use parametric method, and you use this particular mean plus 1.645 multiplied standard deviation, and that give you 95% confidence. For the confirmatory, it's done in the presence of drug, so it's specific. So you basically do the percent inhibition, how much binding is inhibited in the presence of drug. Again, you exclude outliers, you evaluate distribution. If the data is distributed normally, you go with the parametric method, mean plus 2.33 multiply standard deviation, and that give you 99% confidence with 1% false positive. And if the data is not normally distributed, you go with non-parametric with 99 percentile. And they want to have 1% false positive rates because they want to know what's going on in the border population, what's happening in there, which are just right on the border. Outlier removal, analytical outlier, biological. Analytical outliers removed based on residual, and that's the difference between individual results and the median predicted value. And biological outlier removed based on median estimated value for each subject. You have to be very careful when you remove outliers, because outliers should make sense statistically and biologically. The data must have biological significance and not only through the statistical procedures. The RA population has many outliers by the box plot, and it's because RH factor is creating a lot of interference. So you have to know which population you're using. If there is some interference factor, they have to be taken in account. So what we do is we take the data, we put it through the stats. Once we find the outliers, we take them out once, and after that we just keep it there. So even if something is above normal, it's okay. It gives you enough biological variability to have a solid cut point, and you don't end up in artificially lower cut points and have a lot of false positives. That's the distribution, data distribution. For screening, you use 5% false positive rate. For initially, back in 2014, people were using 99% to rule out all the false positives in confirmation. But then FDA changed its guidance in 2016. Now they want at least 1%, and they want, it's okay to have few false positive, but it's not okay to leave any false negative. So that's why they kind of increase their net, and they want to see what's going on. So 1% false positive is fine, even in the confirmatory test now. The titers is the same assay. You serially dilute the positive material, and titer is 1 upon dilution, where the sample OD equal to cut point OD. And you report it with the multiplication of MRD, minimum required dilution. So if your endpoint titer is 100 and you are diluting your sample 10 times, it's going to be 1000 titer. And it just gives you a sense, relative sense of how much antibody is there. By no means it gives you exact amount in nanogram per ml. That cannot happen in AD assays. These are some of the consideration. Screening cut point and confirmatory cut point should be set correctly. Data set accurately reflect biological variability. Enough of the normal range biological and analytical background noise included in the CP calculation, because human population is very, very diverse. People react differently. So there should be enough noise in there. If data is very uniform, you're going to have a lot of false positive. Remove soluble drug targets if they have interference, and you have to balance interrelation of sensitivity, drug tolerance, and cut points and clinical relevance of high ADA positive rates. One downside of this thing is FDA want to have a very sensitive assay who has very good drug tolerance. It end up in detecting a lot of ADAs. The question is, are they clinically relevant? There are ADAs, we can look into nanogram per ml now, but are they doing anything as far as the product safety is concerned? So that goes with the clinicians. They have to see the immunogenicity data and interpret it with the PK and efficacy and safety data, that if it's really going on, are we seeing something, is it relevant? In the end, I would like to say that the opportunities in regulatory sciences, so big pharma, we don't do sample analysis in-house because there are so many samples, tens of thousands, and it's going to be very expensive. So we always outsource it to contract research organizations. We call them CROs, and they're always looking for scientists and analysts. So once you have if you are freshly graduated, it's a good, I think it's a good opportunity. If you can get in there, you can get trained in doing the assays in a regulated way. First, they can train you, and it's gonna give you a special status. You will be part of like a special team now. Anybody can run assay in the lab, but very few people can do it in a regulated manner. So it's a valuable skill to have, and it can help you out. You can get certification and degrees in regulatory sciences, and all the big pharma hires them because they have to make these immunogenicity reports, immunogenicity summaries. So there is a good chance you will get opportunities in big pharma. These are some of the links. I have no affiliation with them, but these are just for the resources. The regulatory affair professional societies, raps.org, they do a lot of certification and degree courses here in US and in Europe as well. US FDA, all the drugs which are in the market, they have their summary available at drugs at fda.gov, and all the guidance documents, how it should be done, they are available here. Same thing with EMA. And then there is International Council for Harmonization of Technical Requirement for Pharmaceutical for Human Use, ICH. They are working on harmonization of the strategies. What will they do in Europe? What do they do in US? So they are coming up with the same terminology, because everybody wants to use their own, and it gets very confusing. So these are some of the interesting, I think, resources you can check out, especially for the students, if you are looking to break in the GMP and GLP business. In summary, I would say that biotherapeutics may elicit anti-drug antibody response with serious potential consequences. Regulatory expectation must be followed as per the EMA and FDA guidelines. The key challenges to overcome when you develop an assay is sensitivity, drug tolerance, and target interference. So a lot of careful thought, a lot of developmental activities, and the data should be interpreted in the really good. And these questions has to be answered well in advance. And cell-based assay, which are the NAB assay, they have a reputation of highly variability due to multiple factors. So a lot of careful consideration, a lot of planning should be done to get these assay working. Thank you very much. If you have any questions.
Thank you, Dr. Vinay, for covering your topic very comprehensively, and in fact giving a big insight into the topic which you just had planned, and covering all the aspects, in fact. And our two of the scientists have put forth a question. So first I'll invite Dr. Vivek Singh to put his question. Dr. Vivek.
Good evening, sir. My first question is, sir, you said that immunoactivation is good for virus, but while treating viral diseases, generally we try to potentiate the immunity or immunomodulation just to boost up it. So whether we are doing right or wrong?
So that really depends. So as I say, HIV, in case of HIV, it lives in gut, in GI tract, gastrointestinal tract. And since it messes up the tight junction proteins, and it basically breaches the permeability. So colon has all the bad bacteria, and the gut bacteria is different. So the bacteria from colon, where your waste is kind of stored, if they entered in your colon, they're going to do a lot of trouble. And as soon as there is cell activation, virus will enter them and replicate. So cell activation and virus replication, they go hand in hand in case of HIV. So that's why these patients have a lot of trouble. They always have diarrhea and they have horrible kind of lifestyle. So if you can somehow lower down the inflammation, if you can close those tight junction proteins, so there is no translocation of harmful bacteria from colon to gut, it would be really helpful. And we have proven it in monkey model that it really helps.
Thank you, sir. Another question is there, sir. Is there any successful transmission blocking vaccines available anywhere against any pathogen?
I think they were doing for malaria as well. There was a Dutch group from Europe, and they were trying to do. I don't know now if they have it. It's a good concept. It's a very public health kind of approach, and the reason is that you can go to a. It's endemic. It's very focal. The transmission of malaria is like in particular pockets. So you go there and you immunize the whole population. So everybody's protected. So if the mosquito take a blood meal from a person who already have the antibodies, the mosquito will not get sick. And if mosquito is not sick, he will not spread the disease. So they were working on malaria as well. So I don't know where are they now. I'm not following them. But there was an effort from Europe to make transmission blocking vaccine for malaria itself. And I think it's very good.
Next question is from Dr. Sanjay Kumar. And in fact, his question is very pertinent to present day requirement. In the COVID era, we all are looking for strengthening our immunity. Dr. Sanjay, you are there? Yeah, go ahead, please.
Yes, sir.
Go ahead.
Sir, my question is in current scenario, the pandemic of the COVID-19 is going on. So what are the easily available modulo activators, immunoactivators are available in the market, which we can access very easily from the market?
So it's a kind of double-edged sword.
Hello.
So, yeah. Hi. Can you hear me?
Yeah, please. Yes, please go ahead.
So it's kind of, you have to be really careful about it. And actually, Nectar applied for a new drug against COVID as well to FDA, but we were denied because there is a cytokine release storm. Cytokine storm could be there, and our IL-2, it's immunoactivator. So you have to be really careful using immunoactivator if you are getting infected with COVID, because if you have COVID and you are having, you can really overburden your immune system. So I don't know exactly what they are prescribing there. I think the best course of action is that just stay home, stay safe, because I don't think there is any vaccine available so far. There are few combinations which they are trying, but I don't know exactly how efficacious they are. Gilead came with remdesivir, and it does help people in serious conditions, but I think it's best to avoid to take the drug and take all the necessary precautions not to get it in the first place.
Fine, thank you, sir. Dr. Saroj, over to Dr. Saroj.
Thank you, sir, for your nice presentation. Sir, this was very informative, interesting, as well as useful for the clinicians. Sir, even though I am from non-clinical side, but it was quite interesting for me. And I hope that our faculty must have gained some information from this presentation. And I am thankful to our Honorable Vice Chancellor, sir, who has spared his valuable time for this webinar. And I am thankful to organizer of this event, Dr. Ravindra Kumar, sir, our Director Research, who has...
Dr. Saroj, we may have the concluding remarks of VC, sir.
Yes, sir.
Saroj, it is fine. I'll take over from here. And today, thank you very much for making a very detailed, exhaustive presentation on the regulatory side of new drugs, and especially the immunogenicity aspect of new drugs. These safety of drugs is of paramount importance, and you said new drug development is very costly. So in silico testing and modification and the kind of protocols you discussed, they can give a good insight to our young faculty and also to our students. that how much science goes into the development of new drugs, and what the chances are there that, as you told about your experience of malaria vaccine, you were very close to it. You really you did eureka in the lab, and finally did not work in the field. So how to take such failures in your stride, and how to learn from all this. You very well said that you got some good reports, good publications out of that, and ended up with all those publications rather than a successful product in the market. So these things are a part of the research career, and the from this experience, the young scientists should develop the culture of science. It is not that we have a hypothesis and we will always succeed. Things would have been very different if we have such a high success rate. Somebody was talking to me that I can tell a paper from an Indian author and a Western author by seeing the conclusion. Many times in India, we feel that I have solved everything, and I will be very positive in my closing remarks that this technique developed by me is excellent and it will solve everything. So we should really see the practical aspect of it and address the issues as they really happen, and also leave scope for further refinement or further work in that. So it has been very informative to me as well as to my colleagues, I guess. And after a long time, I remember Dr. S.R. Anand in NDRI, who used to take a 2-hour class at a stretch. So that was one experience. So he will ask in the beginning whether you want a 1-hour lecture 4 days or you want 2-hour lectures on 2 days. It was a 4-credit course. But all the batches used to go for this 2-hour lecture, and that was really something which you will get a lot of information, and then you have to do a lot of work to digest it. So we are all going for dinner now. And we'll try to digest whatever you have given to us during this more than 1.5 hours of your very loaded presentation. One point I just wanted to find out from you, that a lot of monoclonals are being used nowadays. And what is the frequency of hypersensitivity or chances of immunogenic allergy type of reactions or tolerance as such to the monoclonals which are in the market?
So I guess it really depends on the product you are using, one is using, and generally they give it in the information in the insert, the product insert. They have to say it on the label. So the paper comes with it, they have to disclose the allergy information. But I think MABs are kind of unique, because once you take the MABs, they stay in your system for very long. For example, for our IL-2, for example, for these interleukins, they go out very fast. They work very quick and they get out of the system. But MABs hang around. And that's why I was saying that the trouble with the drug tolerance is basically MAB problem, because they are always there in your system. So since they are there, and I think when you compare with other biologics with MAB, they have a significant chance of the allergy. But in the end, it has to be done like product to product basis. There is no, I cannot really comment on the in general that how they rack up with other biologics.
One for everybody, maybe people are interested to know about that. I read about inhalant for insulin.
Insulin, yes.
Our company has developed a product which you can inhale rather than injection, and it will work for the diabetes or sugar control. How the product is doing, whether we have this product in India?
I think it did not do very well. It was kind of underwhelming response from the markets. Other people kind of took the lead in it.
That
But
was the whole idea, and it is very fast acting as far as compared to the injectables.
Exactly. And I think people who are kind of scared of the needles, it was very good for them as well.
Right. Pricking every time is not good.
Now they have these kind of small device which basically are inside your system. They monitor your glucose level in real time, and they
implants.
You can hang it outside as well. Actually, you can carry a small device, and it automatically check the sugar level, and it can put the insulin dose which is required. So they are smart devices now for diabetes management.
So thank you, Vinay. Thank you very much. We all profusely express our gratitude to you, and you have stimulated the kind of thoughts. People have got the ideas, and I think those who are interested, they can get in touch with you for more information. We'll share your presentation as it was live. It will stay on our Facebook and also on our YouTube channel. And see you again.
Sure.
Have a nice time.
Thank you very much, sir. Thank you very much. The pleasure and honor was all mine. It was great to chat with everybody. And please, feel free to reach out if you have any questions. And I'm sorry if it was too long. I just wanted to touch all the necessary points. It's a massive big subject.
All the participants also. Thanks to all the participants and colleagues who stayed there. And I will again say that you woke up at early morning and took time before your breakfast.
Anything for you, sir. You ask for it, you're gonna get it.
Have a healthy, nice breakfast.
Thank you, sir. Thank you very much. Thank you, everybody. I really enjoyed it, and please feel free to reach out if you have other questions. Thanks a lot, sir.
I'll be in touch with you, Dr. Vinay, and get back to you. Thank you very much.
Sure. Thank you very much. Namaste. Stay safe. Bye-bye.
This session here. Thank you.
Okay, sir.
Thank you. Thank you very much.
Bye-bye.